Awesome. Well, thank you everyone for taking some time out today to come to my talk. I'm hopeful everyone is nice and relaxed and was able to get a massage from the wellness fair that's going on outside right now. If not, I recognize I'm cutting into your free massage time, so make sure you grab one after the talk. But my name is Ericka Schaeffer, and I'm a postdoc in Dr. Burke's lab. So today I'm going to be presenting on some injury-specific effects that we're seeing in peripheral nerve repair following Schwann cell-derived exosome treatment. So peripheral nerve injury accounts for around 3% of trauma cases. Typically, these traumatic injuries can occur from a variety of circumstances. Motor vehicle accidents, gunshot wounds, boating accidents are pretty prevalent down here in Miami. So millions of patients are left with severe sensory motor impairments that unfortunately can be permanent. And severity is all based on how much and which structures of the nerve are damaged. So here we can see an image, a schematic of peripheral nerve. So our axons are ensheathed in myelin. They're grouped together, a bunch of axons into fascicles, multiple fascicles grouped together, held together by the epineurium, make up our peripheral nerve. And depending on which structures of the nerve are damaged, we classify based on injury severity. So again, less severe injuries, axons remain intact, but potentially surrounding structures are damaged, or vice versa, where the axon may be transected, but surrounding structures may be intact. Damage can be so severe that segments of the nerve may need to be, are actually destroyed, and we call this a grade 5 injury, typically occurring from like ballistic or lacerated circumstances. On the other side of the spectrum, we also have peripheral nerve crush injuries that actually can range from all of these different grades here. So it's more of a mixed and variable bag. Typically, we do see crushes land between the 2 and the 4 grade, occurring from a blunt force. Now, following peripheral nerve injury, there's a cascade of responses that are initiated. So there's increased permeability to the vascular system to help recruit inflammatory cells like macrophages. But 3 main processes must occur in order for repair to happen. So first, as we probably all know, Wallerian degeneration must occur. So we're getting rid of those damaged segments of the axons, clearing out that area. And next, axon regeneration must be initiated. And this is an essential function of the Schwann cell. Schwann cells actually change from this myelination to repair phenotype. And they go through this mass proliferation and create the bands of Bunger, which help guide that growing axon to its distal end, help guide that growth cone. Finally, end organ reinnervation must occur. So where the damaged nerve is not only has to meet that distal end, but also function with that target muscle or organ, right? So as we can see, there's a lot of processes that must occur in order for the nerve to, 1, regenerate and also be able to restore function. So there's several considerations we have to think about when we're trying to ameliorate peripheral nerve injury. So let's start with less severe injuries when discussing the current barriers or challenges that must be overcome for successful regeneration. First, scar formation can occur at the injury site. So this could be a physical or a chemical barrier, essentially like a roadblock that won't allow those axons to reach that distal end. So again, disrupting the alignment, disrupting the alignment of those regenerating nerve fibers. We also have target tissue atrophy. This is a big one. Without nerve input, target tissues like muscles or sensory organs undergo atrophy and can lose their ability to respond to innervation. So even if, again, our nerve reaches that distal end, we might have so much target tissue atrophy that we're not able to restore function. So again, limiting that functional recovery. We can also have Schwann cell atrophy. Again, as I just mentioned, Schwann cells play a critical role in regeneration. and secreting growth factors, very important. However, over time, Schwann cells can lose their regenerative phenotype and undergo atrophy, diminishing their ability to support nerve regrowth. Additionally, there's misdirection of axons. So axons often lose their way, leading to misdirected growth that results in inappropriate or incorrect target reconnection. So, for example, sensory axons growing or innervating into muscle sites. So mismatching, again, is going to impair proper functioning. Lastly, for crush injuries, we have some limitations when it comes to current available treatments. Sometimes with crush injuries, it's more of a wait and see. E-stim can be applied. Exercise intervention is also common. But there's a lot of room for growth when it comes to treatments for crush injuries. Now, more severe peripheral nerve injury has these limitations in addition to others. So for example, there is limited regenerative capacity. So regeneration is inherently slow, around 1 to 2 millimeters per day. This is really insufficient to cover long distances to avoid that downstream degeneration of those target tissues and Schwann cells, all the things that we talked about. There's also this thing that we call a critical gap length, where it is the capacity that a peripheral nerve can regenerate. In humans, we typically see this around 4 centimeters. In rats, it is around 10 to 15 millimeters. With large gaps, we can also lose the bands of Bunger. Again, bands of Bunger formed by these Schwann cells to help guide growing axons. However, if regeneration is delayed, these structures can degrade over time. Again, it's more about a timing thing for innervation. Lastly, we have limitations when it comes to treatment. If it is a simple transection, a simple suture repair is most optimal, so suturing the proximal and distal ends back together. But once we have gaps or large sections of the nerve that need to be removed, especially when it's over that critical gap length, grafting must be implemented. So our gold standard procedure is autografting. So this is when we're sourcing a donor graft from the same patient and placing it into that injured area. This typically requires, unfortunately, an additional surgery, could have loss of function in that donor area as well. And there's also some limitations when it comes to autograft. So you only have so much sural nerve to go around to graft. So if you have gaps that are larger than how much nerve you have to spare, you're kind of out of luck there. Allografts are from donor nerves, and if they're not decellularized, they can obviously elicit an immune response, potential graft rejection. Patients would have to be on immunosuppressants, so again, not a real good option there. That's why there's been so much interest in developing these nerve guidance channels or conduits. However, empty conduits on their own don't really promote regeneration or recovery as good as our gold standard, which is kind of what our baseline should be as we're creating treatments. So research has focused on how can we make these conduits better and implementing the use of cellular therapies. So the most obvious choice to supplement recovery of peripheral nerve injury would be to implement the use of cells critical for regeneration, Schwann cells. Early in vitro work from the Miami project found that we can actually isolate Schwann cells from human donors of peripheral nerves. And then once isolated, we can actually implement them into those conduits, into gaps of peripheral nerve injury. First, we have to make sure that we're able to load these conduits properly with the Schwann cells. Several studies have found success in loading conduits with these isolated human Schwann cells, which we can see from here, a slice of a conduit with Schwann cells labeled in red, so we're able to successfully load these conduits. And actually, after 6 weeks of being implanted in vivo in a rat, Schwann cells are still able to be visualized. Which again, at the lower panel is the stained in red here. As for outcomes, when we're talking about large gaps, so 15 millimeter gaps in the rat above that critical gap length, we can evaluate myelin with tubulin blue pictured here. Consistently, we find that conduits loaded with Schwann cells have increased myelinated axons and also thickness compared to conduits that are empty or filled with serum. So the difference between these two studies that I have displayed here is that they're testing out different versions of conduits. So the one on your left is first generation, where the one on your right is second generation conduit, where it has that internal matrix guiding those axons to that distal stump. Interestingly, with the use of the second generation conduit with that internal matrix, we don't find significant differences between the conduits filled with Schwann cells and the reverse autograft. Again, that's our gold standard. As with the older version, the reverse autograft is still the most effective treatment. As for functional outcomes, sciatic functional index was found to be better in the conduit and Schwann cell group compared to the empty conduit and the autograft group, which was interesting. Some electrophysiological characteristics of the gastrocnemius and the tibialis anterior muscle. Action potential onset latencies were faster and amplitudes were higher, were larger compared to the empty conduit groups. So these promising results in rodents in vivo led to implementing the use of Schwann cells in non-human primates. So this study had 3 groups: autograft, empty conduit, and conduit filled with Schwann cells. Improvements were noted in functional outcomes, so in reaching task. Also found increased myelination for the conduit filled with Schwann cells compared to empty. Based on this success, Schwann cells moved into clinical trials to supplement autografts. So in nerve transfers and grafting, success is completely dependent on the donor nerve state. So autografting is not a sure thing, right? You want to make sure that you're optimizing that graft. So supplementing grafts with cellular therapies is a great way to do so. So in this case study, this patient had a very large, severe sciatic nerve injury. I believe it was 7.5 centimeters. So again, well above that critical gap length. Segments of the patient's sural nerve were dissected and used to culture their own autologous Schwann cells. A second surgery was performed to graft that sural nerve into that damaged sciatic nerve area with the supplementation of those autologous Schwann cells. Short-term outcomes found continuity of the sciatic nerve, which is great. Again, end organ reinnervation to that distal end. Long-term follow-up at 36 weeks found functional recovery improvements. So the patient was able to have foot plantar flexion and also knee flexion at 36 months post-op.
But are they absolutely identical or they do differ?
That's a great question. To my knowledge, they're pretty similar within the peripheral nerves. Phenotype is interesting though, because to my knowledge, and correct me if I'm wrong, we don't actually test what phenotype they are as they're being cultured. However, when they're implemented into injury site, the idea is that they will become that repair phenotype.
Ericka, could you just...
The use of Schwann cells in the clinical setting has been successful. There still requires, a second harvesting surgery. There could be immunogenicity if, we can't just transfer Schwann cells from patient to patient. Again, we don't want to go on immunosuppressants. So recently, the Miami Project and others have focused on extracting those therapeutic properties of Schwann cells that won't elicit an immune response. So a variety of cell types, including Schwann cells, release these membrane-bound extracellular vesicles termed exosomes. Exosomes contain microRNA, messenger RNA, and other trophic factors. And these exosomes can be internalized by neighboring cells. So they have the ability to alter genetic information, protein production, they can change cellular phenotype, they can regulate cell-to-cell communication, initiate, terminate, a bunch of different processes that occur. So again, they can have quite a large effect on the cell. Now, there's been promising work done in exosomes as a therapeutic intervention for peripheral nerve injury, including supplementing conduits with exosomes from various cell lines and the use of them in crush models. Our lab and others have implemented the use of Schwann cell-derived exosomes in large gap and crush injuries in rodent models. Specifically, our most recent study, we loaded conduits with rat Schwann cells or human Schwann cell-derived exosomes following a 15-millimeter injury. So what we found is histological and functional outcomes were similar to the gold standard treatment autograft. Although our lab has not assessed Schwann cell-derived exosomes in crushes, others have. So this classic study by Lopez and Varelli performed a crush injury and injected Schwann cell-derived exosomes every day for 4 days post-injury. On the 4th day, they sacrificed the animals and did some histology. And what they found was that the rats that received that exosome treatment had increased regeneration of the sciatic nerve, which we can see from that bottom panel there. So based on the current literature, there's a lot of room to explore. So for example, would we see success in autologously derived Schwann cell exosomes? As the last study that I mentioned, we use human Schwann cell-derived exosomes. Would we get that same effect if we use rat-derived Schwann cells or exosomes? Additionally, much of the crush literature uses Schwann cell-like cells. So these are stem cells that are differentiated into a Schwann cell-like cell, and then exosomes are filtered. What about assessing different injury severities longitudinally, as this really has not been done. And also typically crush studies have pretty short-term endpoints. So our study that we just wrapped up that I'll be focusing on today had 5 groups, first of which was, again, our gold standard treatment of a 13-millimeter autograft. Essentially, we transect 13 millimeters of the sciatic nerve, flip it at 180 and suture it back into place. We also had a 13-millimeter transection group where they received no repair. We had our conduits that were filled with our Schwann cell-derived exosomes. Again, that was implanted into place. And we also had 2 different crush groups. For our crush, we exposed the sciatic nerve and with unserrated forceps applied force for 10 seconds, released for 10 seconds, and repeated that 3 times. So again, a total of 30 seconds of force was applied. And then exosomes were injected into the surrounding area of that injury, then close back up. Here's the timeline of that study. So we did baseline behavior, performed that injury, collected quite a battery of different behavioral assessments. And then week 11 was the endpoint of the study where we did histology and electrophysiology. So the first behavior I want to talk about is motor, which we did a gait analysis. So essentially we filmed the rats from the ventral view during walking. And we'll see, break up my talk a little bit with some rat videos here. So here we have a transection rat and a crush rat. The red arrow just indicates the injured side at week 1, and then also at the endpoint of the study here. Now they're moving pretty fast and it's a little bit difficult to see, but essentially we tracked the limbs to derive different kinematic measures. And from this ventral view, we can also look at more common sciatic functional index metrics. So first I'll start off with SFI, so sciatic functional index, where values closer to 0 indicate better function. Here we found a significant group by time point interaction. So between week 3 to 11, only the autograph and the conduit group had significant improvements in sciatic functional index. The transection and crush groups did not display improvements in SFI across time. However, one thing to note is that the crush animals really didn't have much for improvement, right? They were already, pretty close to zero, so I'm not too surprised that we didn't see that improvement over time for those crush animals. Next on to our gait analyses. The best way to compare across these different injury severities is by using the own animal's uninjured limb as a control. So therefore, majority of the analyses that I'm going to be sharing today are going to be represented by percent difference. So the greater the percentage, the greater differences between the injured and uninjured limb. So during walking, gait is typically divided into two phases based on speed. So first we have our swing phase when the limb is actually in movement, and our stance phase where the limb is stationary. And here we can see a sequence of swing and stances. So swing phase is in red, stance phase is in black for a representative rat during a trial. And the black dots represent the location of the peak speed of limb movement. Typically, that peak speed occurs at the midpoint of the swing phase. Now, since the limb really does not display any meaningful kinematic changes through the stance phase, only the swing phase was analyzed further. So looking at distance, the distance that the limb is moving during that swing phase, we didn't find any effects, but we did some exploratory orthogonal contrasts. So the transection group in purple was found to have overall greater differences between the injured and uninjured limbs compared to the autograph group in blue, which is a little hard to tell, and also the conduit group in green. Additionally, the transection group was the only one found to increase the difference between their limbs from the first time point to last. So again, instead of closing on that difference between limbs, it seems to be getting more divided. Other general characteristics of gait include peak speed, which we found a group effect here. In general, there were greater changes in peak speed between the limbs for the gap groups. So autograph, transection group, conduit group compared to the crush groups in black and in red at the bottom. Additionally, contrasts were conducted to evaluate group changes over time. Across the duration of the study, only the transection group and autograph group displayed greater differences in peak speed between those injured and uninjured limbs compared to all other groups. Now on to more specific kinematic metrics. Now this is an interesting way where we can kind of get a window into the integration of sensory and motor information processing. So typically during the swing phase of gait, limbs make direct ballistic trajectories. And we're able to quantify just how direct is that limb moving during walking. Specifically, we can look at this as a ratio between the actual path taken by the limb and the Euclidean distance, or that shortest distance between that start and stop position. So here we have two representative limb paths, one more direct, if you can see in red, and another one more circuitous in green. The Euclidean distance is plotted by those black lines. So you can see one is making more of an S path, where the other one is making more of a direct trajectory. So in general, rats were made pretty direct paths during their gait across all groups. And typically when animals are accurately estimating distance with their limbs, again, peak speed will occur at the midpoint. So if I'm moving my arm from point A to point B, the location of the peak speed of my movement is going to occur at the midpoint if I'm accurately estimating the distance. So we characterize the location of peak speed during gait as peak error, or peak speed error. The difference in peak error values between limbs varied by group and across time point. In general, differences in peak error appear to be similar across time for all groups, with one exception. The autograph group in particular, so the autograph group in blue, displayed greater differences between limbs from week 3 to week 11. So again, having less similarities between the limbs for peak error. Additionally, at the final time point, so at week 11, the autograph group had significantly larger deviations in peak error compared to the conduit group that was loaded with exosomes. So in general, no differences between crush groups, larger deviations in gait related to distance and speed between limbs for transection animals, and the autograph group estimation of limb movement seems to be intact, but could be affected by speed. So moving right along, the gastroc, Nemesis muscle weight, and some electrophysiological characteristics were compared between injured and uninjured limbs at the conclusion of the study. Regarding gastroc muscle weight, there was an observed effect of group. Essentially, the crush groups had the most similar gastroc muscle weights comparing that injured and uninjured limbs. So again, this is trying to get at that muscle atrophy that we talked about in the beginning. No difference between those crush groups. Next was autograft, then conduit, then transection in terms of muscle recovery. Again, target tissue atrophy, even though those axons are connecting to that distal end, we still had significant atrophy, likely due to time of reinnervation. As for the electrophysiological characteristics, the transection group was not included, as no signal was observed due to not continuity of the sciatic nerve. So we didn't find any effective group with muscle action potential onset or amplitude. Next, we evaluated sensory outcomes. So Hargrave's test was used to evaluate heat sensitivity. We found heat sensitivity fluctuates across time, but there was no specific differences between groups with that. Von Frey was used to evaluate mechanical sensitivity. No main effects were observed here. However, we did look at some exploratory contrasts. The crush and exosome group was found to have reduced difference between the limbs throughout the duration of the study. The remaining groups did not show that similar trend between injured and uninjured limbs. Acetone was used to evaluate cold sensitivity. Nothing was found here in relation to our cold sensitivity test. So in general, nothing really going on, nothing that we could see and really pull out in terms of sensory recovery. Lastly, we evaluated myelin with tubulin blue. So here we can see some representative graphed images of the crush control, crush exosome group, autograph, and conduit exosome group at 4x, and then the axon and myelin rings at 100x. Again, the transection animals were not included in this analysis as that nerve was not present. So the first thing that we found here was a group effect related to the width of the epineurium. So specifically, the autograft and conduit group displayed thicker epineuriums than the crush groups. No differences were noted between the autograph and conduit group. In addition, the ratio between the epineurium's thickness and graft diameter revealed a significant group effect. So essentially, both the autograph and conduit group, again, had greater epineurium thickness in relation to graft diameter compared to those crush groups. No differences were observed between the autograph and conduit group. Next, the number of myelinated axons were found to significantly vary between groups. Specifically, the autograph and conduit group had more myelinated axons than both the crush groups. The autograph and conduit group did not differ in myelin count, and the crushes did not differ in myelin count. However, we're likely seeing these group differences there because of just how large those axons are in those crush groups. I mean, it's pretty compelling by these representative images. So that could be as to why we saw that those crush groups had less axons, is because that they were larger. Axon diameter in relation to myelin thickness was quantified as a G ratio, found a significant effect of group here. Essentially, we found that the conduit and exosome group had the smallest G ratio, which essentially means they had thicker myelin in relation to axon diameter, so a sign of good myelination. So in sum here, again, no differences between those crush groups. Autograph and conduit exosome group, pretty comparable to each other. Autograph and conduit group had more myelinated axons than crushes, but again, could be related to axon diameter there. So what can we conclude from all of this? Well, first, we validated that rat Schwann cell-derived exosomes in large gap are effective. Transection group in general had worse outcomes, and the autograph and conduit exosome groups did pretty similar. So we replicated our previous work in human Schwann cell-derived exosomes. So this is good to know for translatability purposes. Unfortunately, though, we didn't really see a therapeutic effect of Schwann cell-derived exosomes in our crush injuries. Now, this fails to replicate other labs' work who did the same injury type, and this could be due to a few different things. Administration could be critical, so should we have done intraepinurium injections, IV injections, potentially loaded a hydrogel in order to keep the exosomes at injury site for longer, or do multiple administrations. This could also be related to the severity of crush. I tried to look at like a review paper or some article where, okay, this model of crush is equated to this grade of injury, but unfortunately I couldn't find anything. Really what it would be nice to follow up is to reperform this crush injury and then do histology immediately to see what structures are actually damaged, and so we could actually give a grade to the injury that we performed. So it's possible that our crush maybe was not severe enough. It's also possible, it would also have been great to do a no injury control, because these crush injury animals did perform pretty well. They had great sciatic functional index, great walk, no issues with sensory, good myelination. So it would have been great to do a control there, which again is another reason why we use that control limb in our stats. This study was still great in a sense because it generated more questions about exosome-based therapy. So for example, how does administration route vary the duration of exosomes into injury site? And are multi-dose treatments required? What about other injuries? So small gaps, or looking at different severities for crushes, or more of a chronic injury like chronic constriction. So based on this, we have our next study in the pipeline evaluating these exosomes in a neuropathic pain model, chronic constriction injury. So we would like to use in vivo imaging systems to evaluate the internalization, the retention, and the distribution of exosomes after administering them intravenously or injecting around the injury site, like what we did with this previous study. Then we can evaluate neuropathic pain, gait, and actually compare inflammatory markers within the same animal. So do inflammatory concentrations at the nerve differ from the blood, differ from the DRG? So in one animal, getting 3 different sites in order to see, where are these exosomes going, and also what systems are they affecting if we're not seeing too much going on specifically at injury site. So it's possible, again, that our exosomes are having an effect on inflammation, just not resulting in drastic changes to behavior. So I'm actually just wrapping up a pilot study on this, and when I say pilot, I mean like 2 animals per group, so really take this with a grain of salt. But basically, for these few animals, we did the CCI, the chronic constriction injury, waited a week to make sure that they displayed neuropathic pain, and then administered the exosomes IV or injection to the injury site. And then retested them in condition place preference. So the table that you see here, the chronic constriction injury rats displayed preference for the side paired with that analgesiac, that gabapentin, so exhibiting that pain response. However, following the exosome treatment, two IV rats displayed preference for the side paired with saline. So again, flipping that preference, whereas the direct injection group was more 50-50. One rat flipped, the other rat stayed with the pain response. So again, taking this all with a grain of salt, these are only 2 animals per group. We did collect blood and nerves from these animals to run, to compare those TNF alpha levels. So hopefully more to come as this project develops. So much of our future work is based on seeing how we can improve these challenges and barriers in repair for a peripheral nerve injury. So we know timing of repair is critical. We can reduce Schwann cell atrophy with the supplementation of Schwann cells, but how can we make that end target innervation more accurate and reduce atrophy of our targets as well? Also help with the misdirection of axons. This is where we're really interested in combination therapies. So eSTEM has really taken the peripheral nerve world by storm, both clinically and preclinically, great outcomes, increasing muscle mass, increasing regeneration. Therefore, we think that the use of eSTEM would enhance the effect of our cellular therapies as well, more of a synergistic effect there. Additionally, Emily is actually spearheading this with her K. We'd like to do some in vitro and in vivo work to characterize just at baseline specific differences between axon regeneration for motor versus sensory axons. So again, trying to understand that barrier of mismatching that we tend to see in regeneration. And then once we can characterize those differences, how can we ameliorate that? So with that, there are several people who have been critical to this research, including Dr. Burks and Dr. Levy and my other fellow postdoc, Emily. We have an army of medical students and collaborators. So thank you all for your time, and I'm happy to take questions.
How do you decide on the dosing for the exosomes in vivo and in vitro?
So for this specific study, we just did a single high dose study trying to see if there's an effect. The dosage, from my knowledge, we haven't actually done a dosing paper. We're kind of using the highest dose that Yelena can really give us with the exosomes to see if there is an effect. And we're still, especially with the crushes, obviously, we're still not seeing quite a therapeutic effect there. We actually don't, which is one of the reasons that our lab specifically doesn't, which is why we want to implement IVIS to be able to administer those exosomes and actually see that distribution. Where are they going? Yeah. No, actually, thinking about doing the same amount. We do 350 microliters, so we do the same in the surrounding injury area and the same for this pilot study with the CCI intravenously.
Like they're manufactured at the interdisciplinary?
Yeah.
Thank you.
So, I don't know if I missed it. You said you didn't see any differences with the human Schwann cell-derived exosomes either, right?
So we didn't see differences in the human Schwann cell-derived exosomes with the autograft. So that was a previous study that we did, we published last year. We didn't find differences between the gold standard autograft and that human exosome loaded conduit.
They are derived from patients that have no other conditions, and also for every animal you had exosomes from the same donor.
That's a great question. So for the rat derived exosomes, they're actually we just take the same species of rats. So we used Sprague-Dawley's here. Oh, Fisher, yeah, Fisher rats. And Yelena takes those sciatic nerves, homogenizes them, separates out the Schwann cells. So it's the same species, but they don't elicit an immune response in the rats. As for the human Schwann cells, I believe they're from donor peripheral nerves, the specifics of the lineage of that. But I believe there's like 2 or 3 sources, biological sources of those nerves. That is where we derive the exosomes. Not to my knowledge, no. So again, the rats don't have an injury when their nerves are being taken to use for these exosomes or Schwann cells. Oh yeah, go ahead.
Ericka, can you hear me?
Yes.
Oh, great, great. Hey, one more time. Did the animals experience neuropathic pain, and did the treatments have effect on that pain?
Um, so with the injury severity, so with the crushes and the gap, I can't say that we really analyzed neuropathic pain. We didn't do like condition place preference. We just did heat, mechanical, and cold sensitivity with them. And to be honest, we didn't really see anything with that. So from that data, I can't say that they were exhibiting signs of neuropathic pain and or that we ameliorated that in any way.
We've imaged the exosomes after IV infusions, and they go to most all organ systems, and some end up in the CNS, which is interesting. We're now working on with carbon dots to try to enhance the accumulation in nervous tissue. So that's an area of active investigation. So in our early studies where we actually treated patients, in some of those individuals, we saw pretty impressive return of motor function in terms of walking, for example. So, what's your hypothesis in terms of understanding the mechanisms by which uh, enhancing axonal growth across the gap would ultimately lead to improved motor function?
Well, I think a lot of that is in terms of speeding up the time it takes for regeneration to occur because you want to reduce that atrophy of the muscle. I think that's a big component that we are now starting to look at is it's not just about does the axon get to the distal end, but is it reinnervating with a healthy muscle that can then improve function as well. So more of a connection required in order for functional recovery to occur.
So, the regenerating axon does not necessarily have to travel a long distance and make a synaptic contact on the muscle, but possibly the exosomes, especially if you're infusing them systemically, could potentially enhance maybe innervation or function of muscles and therefore improve function?
Yeah, exactly. I think that is definitely a testable hypothesis that we can look at, especially when we know the distribution of exosomes more specifically. We might be able to answer that theoretically.
Yeah, we've now published 2 papers with systemic infusions of human Schwann cell exosomes, and we're going to spend probably the rest of our career trying to understand the mechanisms by which this systemic infusion of exosomes has such widespread effects in terms of reducing brain edema, improving cognitive function, and also dramatic effects on the inflammatory state of the blood, for example. So this is a very exciting area and really exciting presentation today. Thank you very much.
Thank you. Thank you so much. Yep.
Dr. Dietrich's hypothesis is right, and you're just getting an increased rate of peripheral nerve regeneration with the systemic infusion. If you were planning to cut a nerve, maybe to transfer it on to another, would you use this approach as well, or?
I think that there are a lot of ways that you can go. We're actually in our peripheral nerve patients clinically, Dr. Burks is working on doing pre-op electrical stimulation to almost prime that donor area, that donor nerve, and also that target area as well. So I think the best state possible for, let's say, a nerve transfer is most optimal. However, that's not, the case for injuries that don't necessarily require a nerve transfer, like crushes or, chronic constriction, different injuries that don't require that.
They're not elective.
Yeah. Awesome. Well, thank you everyone for coming.